All right, let's just jump right in. Right now, inside your body, there is this incredible high precision construction project going on. Your cells are building and rebuilding these microscopic living zippers that are quite literally holding you together. And today, we're going to see exactly how they pull it off. It's a really simple question, but the answer is pretty mind-blowing. Think about it for a second. We're mostly just bags of salty water. So what is it that stops our trillions of cells, all our tissues and organs, from just leaking? Well, it all comes down to the amazing seals our cells make with each other. And these seals are, well, absolutely essential. They create the barrier between outside and inside at the cellular level. They keep all the good stuff in and all the bad stuff out. I mean, without them, our tissues just couldn't work, period. Now, the structures that do this incredible job are called tight junctions. The best way to think of them is like the Ziploc bag of the biology world, except they're way, way smarter. This is a whole network of proteins that zips neighboring cells together, making a seal that's not only super strong, but also surprisingly alive. Okay, so at the very heart of this whole operation is one key protein, a scaffold protein called ZO1. Think of it like the foreman on the construction site. It doesn't actually make up the teeth of the zipper itself, but it's the one gathering all the parts and telling all the other proteins where to go and what to do. It's the master organizer. And that leads us to the big mystery. How do millions of these individual ZO1 proteins, which are just kind of floating around inside the cell, know how to line up and form a perfect unbroken belt around every single cell? For a long time, this was a huge puzzle for scientists. And the first big clue came from this really cool physics principle called phase separation. You've seen this a million times in your kitchen. You shake up some salad dressing and the oil and vinegar just naturally separate out into their own little droplets. Well, it turns out our cells use that exact same trick. ZO1 proteins can spontaneously clump together out of the cell's watery interior, forming these tiny, liquid-like droplets. And this isn't just a theory, A 2019 study published in the journal Cell saw this happening in real time. Scientists literally watched as these little clusters of ZO1 moved towards the edge of a cell and then fused together into bigger blobs. That was the aha moment. It proved that ZO1 really does behave like a liquid. So this brings us to part one of our story, which is all about a groundbreaking 2024 discovery. How do you get from these liquid droplets to a solid continuous belt? Okay, so here's how it works. It starts where two cells touch. Receptor proteins form little clusters, like sticky patches. Our ZO1 protein droplets are drawn to these patches and condense right on them. Now here's the brilliant part. Those droplets then call over actin, which is like the cell's universal 2x4, its main building material. The actin starts growing into these long filaments, and as it grows, it pulls on the liquid ZO1 droplets, stretching them out like taffy. This pulling action elongates the droplets until they're long enough to touch and fuse with their neighbors, creating this perfect, seamless belt around the whole cell. And that 2024 study in developmental cell proved just how vital this connection is. What they did was they created a mutant version of ZO1 that couldn't grab onto actin. And the result, you can see it right here. The whole building process, the actin growth, just grinds to a halt. It was the smoking gun. It showed that ZO1's ability to grab and organize actin is what drives the entire thing. So now we know how the cells build the belt. But that's not the end of the story. Not even close. See, these aren't just static walls. They're alive, and they have to deal with real physical forces in a living body. Which brings us to part two. These junctions can actually feel. That same 2019 study, the one in zebrafish embryos, uncovered something else totally remarkable. These junctions are what we call mechanosensitive. That's a fancy word, but it just means they can literally feel when they're being pulled or stretched. And not only can they feel it, they respond to that force to make themselves stronger. The whole system is incredibly clever. When there's not much stress, a junction has a normal amount of ZO1 just doing its job. But when the tissue gets stretched and that junction is put under high tension, it basically sends out an SOS signal, like, hey, we need reinforcements over here.
the cell immediately gets to work, sending more Z01 right to that spot to strengthen the seal exactly where it's needed. And what's so cool is how the researchers prove this. They actually use these microscopic tools to physically pull on a single junction, creating artificial tension. The result was immediate and obvious. The amount of Z01 that rushed to that specific spot shot way up. It was direct, undeniable proof of this on-demand reinforcement system.
So how does the cell ship these reinforcements to the right place? It uses this beautiful little delivery system. The cell's internal actin network is always flowing, kind of like a series of tiny conveyor belts. These belts pick up those spare Z01 droplets that are just floating around and ferry them directly to the parts of the junction that are crying out for help. It's an incredibly targeted, on-demand supply chain. Okay, so let's put this all together. What we're looking at isn't a simple brick wall. It's this incredibly dynamic, living structure that builds, adapts, and maintains itself in real time. This timeline really tells the whole story. First, you get nucleation, where the Z01 droplets form. Then, elongation, as the actin machinery stretches them out. That's followed by fusion, where they merge into a solid belt. And finally, you have this constant process of reinforcement, as the cell's conveyor belts bring in more material to any spot under tension. It's a nonstop cycle of building, sensing, and strengthening. So why does all this matter? Well, it totally changes how we should think about these crucial barriers. They're not static walls, they're dynamic self-healing structures that are born from liquid parts. This ability to build themselves and then strengthen themselves on the fly is absolutely critical, especially when an embryo is developing and tissues are constantly moving and stretching. It's this system that makes sure that as we grow and change, our tissues hold together perfectly sealed. Honestly, it's just a brilliant piece of biological engineering. To build a strong, reliable wall, our bodies don't use solid bricks. They use adaptable, flowing liquids. And that really leaves you with one last thought to chew on. If this is the elegant, completely counterintuitive solution our cells came up with for a simple seal, what other incredible marvels are still hiding, right there in plain sight?